Hello, everybody, and welcome back. I hope you guys all studied hard for your last exam and that you did well. This is the third big section of the semester, so we're over halfway through pretty much at this point. And from here on out, we're just going to continue to talk about the different phyla of animals, and you'll see how they continue to increase in complexity. So the first lecture we're going to have in this third section of the semester is discussing annelids, and this is chapter 17 of your textbook. Let's start, as we always do, with some general characteristics of the phyla. So the phyla Annelida is composed of a variety of different organisms, and the organisms that fit within this category are often referred to as bristleworms. And this is because they have these hair-like bristle projections on the outside of their body that are called setae. This is with the exception of leeches. So leeches don't generally have setae, and we'll talk about what we see in leeches in a second. But just know that these hair-like projections come up a lot and are actually involved in some of the classifications of the annelids as well. Annelids are lophotrochozoan protostomes, and this is because they have a trochophore larvae. Some have lost their trochophore larvae, like we saw in some mollusks have also lost their trochophore larvae, but it is ancestral to annelids. So there is a common shared ancestor that had this trochophore larvae. Annelids undergo spiral mosaic cleavage in early development. And annelids are the first phyla of organism we're going to talk about this semester that exhibits segmentation or metamerism. And the segmentation we see in annelids is actually an analogous trait to the segmentation that we see in arthropods and in chordates like humans and fish and reptiles and things like that. So that means it evolved separately. But essentially, metamerism in annelids, like I said, is body segmentation, and each of those segments of the body plan have similar components of all of the major organs of that organism. And we'll go through each of those organs and the segmentation in the slides to come. The metamerism in annelids is pretty apparent. They're designated on the outside via something called annuli. So when you see a worm, and we'll see some pictures in a minute, I know we've all seen an earthworm. It has those like rings on them. Those are the annuli that you can see on the outside. But segmentation also extends to the inside of the worm as well. So each of those segments is designated by something called a septa. So each segment has its own kind of walls that dictate each segment from one another. And then some annelids have lost their segmentation secondarily, but they all had a segmented ancestor. So this is a synapomorphy of annelids, and then some of them have the derived characteristic of the loss of segmentation. Annelids are coelomates, so they have a true coelom. They develop their coelom via schizochelous coelom formation, and their coelom is also an analogous characteristic to the coelom that's formed in arthropods and humans and other organisms. They also have a compartmentalized coelom, so they have a septa that prevents their coelom fluid from moving between segments. And so what I talked about, there's like a wall that separates each segment. Well, that wall also keeps the coelom that's present in one segment confined within that segment. And then this allows for each segment of the organism to move independently because it has its own coelom, its own hydrostatic pressure. So each segment can move independent of the other segments. Annelids have both longitudinal and circular muscles, and so we'll talk about how that's important and why that's important in the locomotion of the organism in a few minutes. But annelids can move via crawling, swimming, burrowing, and then some are pretty sessile. So this image here that's up here, these tube worms that live near hydrothermal vents, they are considered sessile because they basically anchor themselves into soil and they create this casing and they never really leave that casing. And then annelids can be terrestrial, marine, or occur in fresh water, so they're pretty ubiquitous, pretty versatile. They do need moisture though, because they don't have respiratory organs, and so they require gas exchange to occur through their skin. So they do require moisture, even though they can live in a variety of locations. Some examples of annelids you might be familiar with Earthworms, leeches, and bristleworms are just to name a few. So before we really get into some examples of annelids and their anatomy, there's some terminology I think we should all be familiar with. And this terminology is based in the phylogenetic analyses of annelids. So there's a couple of groups that have come about over time, and I'm presenting them here as old and new terms. They're not necessarily old and new. You see them both now. We use both of the terms that are considered old and new, but there are some issues with some of these older terms that require some refinement, and some of these newer terms are an effort to try to make more valid clades when we're looking at cladistics. So the first term we have in our old category is polychaeta or polychaetes, and these are our worms that have many setae, and they composed of mainly marine worms. The marine worms can live either a benthic or pelagic lifestyle, which either just means that they live on the ground, on the seafloor, which is benthic, or pelagic means they're just free swimming in the water column. But the problem with this grouping is it's paraphyletic. And just as a reminder, when we're trying to do our phylogenetic analyses, we want monophyletic groups. Those are the only ones that we're considering to be valid. So para and polyphyletic groupings, we want to try to get rid of those over time. So that's why this grouping needs some refinement so that it's no longer paraphyletic and we can come up with new groupings that are monophyletic. In addition to polychaetes, another paraphyletic grouping we have are oligochaetes. And oligochaetes just basically means few setae. So they have setae, just not as many as the polychaetes do. And this is composed of most of our freshwater and our terrestrial worms. The third term that I'm presenting here is our worms that fit in the class Hirudinida. And these are your leeches. And this is the only monophyletic grouping that your book kind of presents to us. And this is composed of leeches that live in freshwater and then a few that live in marine environments. So of our new quote-unquote terminology, we start with the worms that are considered Clitellata. They're in this group. And the signature characteristic of all these worms is the presence of a clitellum. And a clitellum is basically just a thickened body segment that produces the cocoon for the eggs and for reproduction in these worms. And you'll see it, and once you see the picture in the next couple slides, you'll be like, oh, I know exactly what that is. I've definitely seen that on earthworms. This grouping is composed of both some oligochaetes and leeches. So you can see here that in an effort to make more monophyletic groupings, we're taking some of those older groupings and trying to get more specific with them, because this clitellum is a homologous character. So it's more indicative of a monophyletic group. And then another term we have is the organisms that are within the group Erantia. Sorry, it's a little challenging sometimes. And these are basically just your free moving, free swimming, free crawling worms. And this is mostly composed of polychaetes. And then finally we have our sedentaria, and these are the worms that basically make either tubes or burrows, and they just stay in there. So from this name you can kind of figure it out. Sedentaria, they're sedentary. And this is composed of polychaetes, oligochaetes, and some leeches. So these groupings are an effort to get to the more monophyletic groupings and to get away from some of the more paraphyletic groupings, and those can just be used as descriptives. So let's start by going through each of those groups and talking about some key characteristics of them. So let's start with Erantia. Erantia, as I mentioned, are our motile polychaetes. These are ones that maybe swim free in the water column. They may be active predators. They may actively crawling. These are the ones that are motile. Most of them are marine, and they can exhibit bright, beautiful colors. So I know many of you probably have seen a lot of documentaries where they have these cool looking segmented worms that crawl around the coral reefs and things like that. Those more than likely fit in this category as long as they're a segmented worm. They have many setae. So I had mentioned these kind of bristle-like hair projections, and that can be one of the best examples seen here is these little white puffs are the setae on this organism. And these setae are on something called parapodia. And the parapodia are a little bit more challenging to see, but there you can see there's the main body that's here. And there's these projections that are kind of like right in these little areas, and they look like little arms. And then off of those projections, you can see the many puffs of setae that are white. Those projections are called parapodia, and these parapodia play various roles in locomotion and in respiration. So let me just draw something here because it might be a little bit more clear. So let's say we have our worm body, and then on our worm body they have these little projections. Those little projections would be the parapodia. And then on those parapodia, some of them have many hair-like projections, and those are the setae. So I hope that's a little bit more clear. They do not have a clitellum, so they do not fit in that grouping. They do have many sensory organs because many of them are predators. And so in order to, actively catch prey, you have to be able to sense that prey and also just be able to sense danger. And then some of them are scavengers. So, if you have a dead fish or something that's died, they'll go along and go ahead and eat it. Some examples are clamworms, scale worms, and fireworms. The fireworm is this example here. And they are called that because those they have projections that will inject toxin into you, which causes a burning sensation. So, but even though they're still super beautiful, but don't touch them. The next big grouping is sedentaria, and sedentaria include some polychaetes and some oligochaetes. And their big characteristic is that they live in tubes or burrows. And they'll live in these tubes or burrows for the entirety of their life, unless something happens like they're about to get eaten or they get majorly disturbed. And their tubes and their burrows, but they make the burrows themselves, or they can inhabit burrows from other organisms. And same thing with the tubes. Many of them make their own tubes through mucus and other compounds that are excreted from the worm itself. But then some of them also will use other organisms and products from other organisms like shells or debris and stick it together using mucus to make their tubes. So in either case, the big key characteristic is they live a sedentary lifestyle. And so they don't leave those tubes or burrows for pretty much their whole life. Many of them have tentacles to capture food and also to undergo respiration. So a good example, it can be seen here. This beautiful organism looks like an undersea plant, or maybe even like an undersea fern, but it's not. It's actually a worm. The worm lives in this tube down here, and then these projections are actually tentacles that it's putting out into the environment to capture food. So they have these tentacles that are covered in cilia, and some of them don't have cilia. Some of them have tentacles covered in mucus. Some have tentacles that are covered with cilia and mucus. In any case, they're all used to capture food. So they're suspension or particle feeders. They put these tentacles out into the environment, and any type of detritus, small bits of matter that will stick to that mucus or stick to those cilia is then pushed to the mouth so they can feed. So this is very similar to what we talked about in filter feeding in bivalves in our mollusk lecture. And some examples are tube worms. So in the first slide, I showed you a picture of those deep sea tube worms. That's one example. Also this fan worm here, beard worms, then spoon worms, which this is a spoon worm, and they're called that because they have a spoon-like projection at the end of their tentacle they use for feeding. Next, we have the group Clitellata, and Clitellata is composed of oligochaetes and leeches, and their key defining characteristic is the presence of something called a clitellum. And an example of a clitellum can be seen here, and it's basically that kind of fat section that you've probably seen if you've ever seen an earthworm that's generally towards the first third or fifth kind of area of the earthworm. And it's responsible for producing some mucus that's later used to make a cocoon. And this cocoon is important for birth fertilization and for kind of protecting the young until they're ready to leave the cocoon as young worms. This clitellum is always visible in the oligochaetes in this grouping, like earthworms and things like that, but it's only visible in leeches when they're ready to reproduce. So you may not see them in leeches if you just see a normal leech unless it's ready to undergo mating and reproduction. The clitellata do not have parapodia, so they don't have those kind of little pseudo feet on their bodies. And this is because they lost it secondarily. So they had some sort of ancestor that did have parapodia, but the derived characteristic for this grouping is that they've lost them. And they have, most have setae, and we'll talk about the leeches in a second because leeches generally either don't have setae or they have very, very few only in certain areas. They are monoecious and they lack a trochophore larvae. So this is also a derived characteristic of this grouping Clitellata. They have an ancestor that had a trochophore larva, but at some point in their evolution, they lost that stage in their life cycle. Instead, their young emerge from their cocoons as fully developed small worms. So basically small versions of the adult. And then the organisms that fit within this grouping can be free living or they can be parasitic. And you're probably like, yeah, leeches, duh, they're parasitic. But there's also some other worms out there that can be parasitic as well. And some examples, earthworms and leeches. And then the last group we're going to talk about is the organisms that fit in the grouping Hirudinida. And these are your leeches. And the leeches are a monophyletic grouping, and there are actually 3 orders of leeches out there. We have our true leeches, and then you have your other 2 orders, and I'm not even going to attempt to pronounce them, but the other 2 orders, their morphology is somewhere between true leeches and the oligochaetes. So they're morphologically different from the true leeches, so they get their own order. Leeches all have a specific number of segments depending on what grouping they're present in. So if you're with this particular species and genus, they will have maybe 37, and in this one they might have 27, but it's conserved no matter what group they're in. They have little to no setae, so most of them have lost their setae secondarily, but some of them do have a few as particular regions. They do have anterior or anterior and posterior suckers. So in this case, you can see in this organism here, it's got an anterior sucker that it's using to stick to this leaf. And you can't really see the posterior region, but I don't think they have one on the posterior. But some of them have one at their anterior end and one at their posterior end that they can use for feeding or anchoring themselves to their host or the substrate. They can live in fresh water, but then some of them can be marine or even terrestrial. So this one is an example of a terrestrial leech that is clearly on a leaf. And then this is an example, you probably can barely see this little guy, of one that's marine that's living on a lobster. So they come in a little bit more variety than the normal kind of river leeches that we always think of. And they are carnivorous and parasitic, which makes sense, right? They use a proboscis for hunting when they're being active carnivores, active hunters. And then when they're being parasitic, they use their jaws to attach themselves to their potential host. And then they use anesthetic to make sure that their presence goes unknown. So they, once they attach to you, they release the anesthetic so you don't really know that you're being fed on. And then they also release a anticoagulant. And this helps for the blood to flow smoothly from your body into their gut. So they have a couple of different mechanisms to optimize their ability to feed, whether it be through active hunting as carnivores or via parasitic lifestyles. The leeches, the group leeches, Hirudinida, is monoecious, as I mentioned before, and they do have a clitellum, so that's why they also fit under Clitellata. But as I mentioned, it only is present when they're ready to reproduce. So I term that as temporal. And then they do have a coelom, but their coelom is quite unique. There are only a few animals out there that their coelom has been filled with connective tissue. And so there's so much connective tissue within that fills their coelom that they only have channels of fluid that run between the connective tissue. So there is a true coelom, but that coelom has basically been invaded by connective tissue. And another cool thing about them is many of them have two brains. So I don't know why. I guess to help with this kind of anterior-posterior attachment, because if they have two brains, they have an anterior brain and a posterior brain. So now that we've talked about some of the groupings of annelids and their diversity, let's get into their anatomy, starting with their body plan. So we're going to start outside in. So the outside of the annelid body is covered in a cuticle, and many of them have these hair-like projections called setae. So earlier I tried to draw a picture for you guys, which is probably a very crude drawing, but here you probably better see them. So these projections here on the parapodia, and these were the parapodia that I was trying to draw, are the setae. And they're aiding in locomotion, but they can also aid in anchoring the organism to its burrow or its home. So this can be important, as you can imagine. If you're an earthworm and a bird is trying to come and eat you, you're pretty defenseless. Like worms, they can only fight back so much. They don't really have, at least earthworms, they don't really have much defenses, but they can try to make it hard for the bird to eat them. So as they're crawling through their burrow, they use these setae to anchor themselves into their burrow. So when the bird tries to pull it out, the worm doesn't just easily slide out of its burrow. It requires a little bit of force because the setae have anchored that worm into place. Many of the annelids, or some of the annelids, have lost these setae, and that is a derived characteristic of those groups. All annelids are derived from a shared common ancestor that had setae. The annelid body plan has a couple of different segments. They has a head, and the head has two parts. And then they have their body, which is segmented all the way down until you get to the pygidium. And the pygidium has the anus. So let's begin at the head. The head, as I mentioned, has two parts. We have our prostomium, which is the anterior region before the mouth. So this image you can't really see so well because it's looking at the top of the organism, but this area here is the prostomium. So where you see the jaws is not, the mouth is not right where the jaws are. The mouth would actually be here in this first segment of the worm, and it's located where that segment we call the peristomium, and this is the first true segment of the worm, and that's where the mouth is. So the prostomium has usually tentacles, eyes, sensory organs, anything it needs to like hunt, but the mouth is actually at the peristomium, so it's a little bit anterior to the kind of where the mouth parts are. And then they have a segmented body plan. This can vary in length depending on the worm. They can be very, very few, up to hundreds of segments. There's some really giant worms out there. So those are a little bit variable. Each segment of the annelid body plan contains key structures of the organs that are required for the annelid to survive. So each segment has its own circulatory, nervous, and excretory structures, and then each segment also has its own coelom, so its own walled-off fluid-filled cavity. This segmentation does not extend to a few of the key structures, and those are being the gut, which can be seen here, the dorsal and ventral blood vessels, or the ventral nerve cord, which you can't see in this diagram, but is actually somewhere in this area here. And instead of each of these key structures being segmented like the rest of the body plan, they actually just run all the way through the organism from the anterior region, so from the head, all the way to the posterior at the anus. So the gut runs straight through, and so do these vessels, and so does the nerve cord. Each segment may also have a parapodia. This is not required for annelid to be considered an annelid, but some of them do have these parapodia to help them with locomotion and help them with respiration. When we want to add a new segment to an annelid, they're actually added at the posterior region of the animal. So we start at the pygidium. The pygidium is here, and it's the most posterior part of the animal, and it contains the anus. New segments are added just anterior of the pygidium, so they're added right here. So this segment of this animal, this worm in this example, would be the newest segment of the animal. And then this segment would be second newest, third newest, fourth newest, and so on and so forth. So if a new segment needs to be added to this worm, it will be added just anterior of the pygidium, pushing this segment right here to now be the second newest, and then eventually the third newest, blah, 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 as the organism continues to elongate and get longer and longer and longer. So this means that the oldest segments of the annelid are present closer to the head, and then the newest segments are located closer to the anus. So when we're looking at the coelom, I mentioned before that the coelom is compartmentalized. So the coelom functions to protect the internal organs from any damage, but it also provides something we call the hydrostatic skeleton to the organism. So worms don't have bones. They don't have a skeletal system in that way. Instead, they have their fluid-filled coeloms that have been walled off that help them to provide structure to the organism so they don't get crushed under the weight of pressure from the water or from soil and things like that. The coelom is, we can look at this diagram here. Let's erase some of this. Okay, so the coelom is located in this image here. And then this wall behind the coelom that I'm just kind of scribbling here is showing you the septa. And then they also have a picture of the septa here in this image right there. And it's basically, that's the wall that separates this segment coelom from the coelom of the segment before it and the coelom of the segment behind it. And then the coelom is also surrounded by this tissue called the peritoneum, and you can see it here. They have two peritoneum. They have one peritoneum that separates the coelom from the muscles, and they have another peritoneum that's out and separates the coelom from the gut, so that it's walled off fluid-filled cavity that doesn't leak out into the gut or into the muscles. So the hydrostatic skeleton plays also a role in locomotion as well. So, and this is in combination with the muscle fibers. So I talked about before that annelids have two types of muscles. They have circular and longitudinal muscles. And the circular muscles can be seen in this diagram right here. And these circular muscles run the circumference of the organism. So they're all the way around the annelid. And then they also have longitudinal muscles that run from the anterior to the posterior of the animal. And both of these types of muscles work in an antagonistic way to one another. So once one is stimulated, the other one is relaxed, and vice versa. And this plays a role in locomotion because as the organism is moving forward, if it wants to make certain segments thicker and shorter, like wider and shorter for anchoring, they can, and without making the whole organism like this. And then certain segments they can make longer and thinner to project themselves forward, and they will activate the longitudinal muscles for this. So this is a bit hard to explain, so I definitely recommend watching the video at the end of this presentation where it shows you an earthworm crawling and you can better see what I'm talking about. But I'm going to try to explain it here. So let's say we have a worm, and our worm wants to move down this page, okay? So it will need to expand, it will need to elongate some sections of the body, and then it will need to expand other sections of this body to anchor itself in place and then project itself forward. So this worm wants to make these sections of its body thicker and wider for anchoring itself into its burrow. So what it will do is will activate the circular muscles in that region, so they become shorter and wider, and they will use their setae to stick into their burrow, anchoring them in place. So instead of looking like this, the annelid might now look something like this. And then in order to progress themselves forward, they need to activate the longitudinal muscles at their anterior region to become thinner and longer so they can inch forward to new territory. So to do that, this section of the organism will need to be activating its longitudinal muscles. So now instead of looking like this, it will look something like anchored in place, and then long and thin, anchored in place, and then the anterior and posterior region. So now it's extending itself forward to reach, and then once it's kind of extended forward, it will need to now activate the circular muscles and relax the longitudinal muscles in this section of the body so that it can anchor that part in place. And then it will also need to relax the circular muscles here so that the rest of the body is not anchored in two places, and the posterior part of the body can be moved up towards the anterior part of the body. So it will now look something like, oh, I'm running out of space. Let's see, posterior region, anterior region, head. Okay. These are very, very poor drawings. I'm trying my best. As I said, I definitely recommend looking at the video of the earthworm crawling. It will make a lot more sense once you see it, how certain muscles, in order to move forward, need to be activated, which are the longitudinal muscles, and the circular muscles must be relaxed. And then vice versa, when you want to anchor yourself in place, the circular muscles need to be activated and the longitudinal muscles need to be relaxed. This relationship between the circular and longitudinal muscles also play a role in swimming for aquatic versions of the annelids. Annelids can be either filter feeders, scavengers, hunters, or parasites, and for the most part, they have a complete gut. So that means they have a mouth, they have intestines that run the entire length of their body, and then they have an anus. This is with the exception of organisms that belong to the group Siboglenids and their close relatives. These are the tube worms that live near hydrothermal vents, and they don't have a mouth, a gut, any type of intestines, or an anus. So it's really interesting to learn about them and how they're able to survive and feed without being able to bring in food through a mouth, process it with intestines, or get rid of waste via an anus. So if you're interested in these tube worms in a little bit more detail, I have included 2 videos at the end of this PowerPoint that talk about them in a lot more detail. So getting back to the vast majority of annelids having a complete digestive tract, the digestive tract of polychaetes has been divided into 3 parts: the foregut, the midgut, and the hindgut. And then oligochaetes, they have some key structures, which are the crop, the gizzard, and the intestines, as well as a mouth and an anus. So some of these structures that I'm going to talk about in oligochaetes are also present in polychaetes, but I found some better images to explain it with oligochaetes. So we start with the mouth, which is of course used to obtain food, catch prey, attach itself to the host, whatever have you. And then food is moved from the mouth through the pharynx and the esophagus, which bring the food down to the crop. And the crop is responsible for storing the food and housing it until it's ready to be moved to the gizzard. And the gizzard is a specialized structure that breaks food down to smaller particles. So we do this by chewing our food. We break down large bites into smaller, more manageable, more digestible pieces, and then move it into our stomach. But worms don't have teeth, thank God, because that would be really creepy. Instead, they use this gizzard to break down their food, larger food particles into more manageable pieces. From there, the food is moved to the intestines for absorption, and then anything that's not wanted is then removed via the anus as waste. Annelids have a couple of different structures they can use to undergo respiration. Some of them have parapodia that they can use in combination with setae to help with respiration. Some of them have gills, so this would be obviously more aquatic forms. Then some have tentacles they can use to undergo respiration. So we talked about this with the fan worms, how they can use their modified tentacles that are covered in cilia or mucus to catch prey, but they can also use those projections to undergo gas exchange. But a big reliance for gas exchange is through the skin. So annelids, like I mentioned before, they have to be living in areas that are moist. And this is because if their skin dries out, they can't undergo respiration and they'll dry out and die. And so they need moist environments so they can undergo active diffusion through the skin so they can undergo gas exchange. Annelids often have respiratory pigments, and these pigments can vary depending on the species. Some have hemoglobin, some have other respiratory pigments that they use. Annelids have two types of circulation. They have their coelomic fluid that can aid in circulation, and they also have a closed circulatory system. So we talked about an open circulatory system in mollusks, how they have a heart and they have vessels that transfer the blood from the heart to whatever organs need oxygen and nutrients. But once the blood gets to its target, there are no capillaries. So the blood mixes with the interstitial fluid that's around the organs and basically becomes hemolymph. And then eventually it is brought back to the heart. With the closed circulatory system, like we saw in cephalopods, they have actual true capillaries and true hearts. So blood will flow from the heart through to whatever organs need it. The blood stays in the capillaries, drops off its oxygen content, and then returns back to the heart. In worms, they have true hearts. They have a couple of hearts, if you will, and they can be seen in this image here as these kind of enlarged aortic arches. They also have two vessels. They have the dorsal and the ventral vessel, as I pointed out before, but in this image, it's the red one here is our dorsal, and then the red vessel here is our ventral. So blood comes in through the dorsal side, goes and gets oxygenated at the heart, and then travels back out the ventral side to go to wherever it needs to go to oxygenate those organs. And the blood is interesting in this case that they also have amoeboid cells in their blood and respiratory pigments, i.e. hemoglobin. For the excretion of waste, annelids have basically two main options. They can either just excrete their waste through their skin, as they kind of do when they're respiring, so they would just use diffusion to remove the waste. But then they can also use something called the metanephridia. And the metanephridia are kind of like kidneys, but they're not true kidneys. And there's one pair of metanephridia for every body segment. So in this image up here, they're trying to show you that there's a metanephridia kind of on this side of the coelom and on this side of the gut. So the gut kind of runs straight through. Actually, this is a good image of it. The gut runs straight through each of the segments. And then you can see in this image, each segment has its own metanephridia. It has two of them, generally. Some may have one, some may have more than one, but generally they have two. So how do these organs work? So the metanephridia has something called a nephrostome, which is this opening. And the nephrostome of one segment's metanephridia actually opens in the segment anterior to it. So it bisects the septum that separates the different segments and opens up over there. So the nephrostome has cilia that bring in the coelomic fluid, and the coelomic fluid will travel through the metanephridia through this kind of maze of loops, kind of like you see in the kidneys of humans. And as the fluid's traveling through, any of the fluid that's needed for the organism will be reabsorbed, and then the waste will be concentrated. Eventually, the waste will get to the nephritiopore, and the nephritiopore is just the opening to the outside world. So coelomic fluid comes in through the nephrostome, travels through the metanephridia, and then leaves via the nephritiopore. The annelids can either excrete ammonia or urea. Ammonia is far more potent than urea, but depending on the species, they may excrete either or some excrete both. And the osmoregulation of the organism is through the skin and through the nephridia. So we talked about osmoregulation in the flatworms and how it was mainly responsible through their kidney-like structures. Similar thing here, but they can also do osmoregulation through their skin. The nervous system in annelids is a little bit more complex than what we've seen so far, with the exception of like cephalopods. So they have a true brain, which you can see here, that's located at their anterior region on their dorsal side. And then they also have a nerve cord that's on their ventral side that runs all the way from their brain at the anterior region to their posterior region. In addition, they also have these nerve nets, like not nerve net, but they have lateral nerves that are located in each segment. And these nerves have neurons that will connect to the muscle fibers and allow for muscle contraction and relaxation so the organism can move. In addition to this nerve tissue, they often have eyes. So annelids can have either a simple or complex eye. They also have statocysts that help them with their orientation and equilibrium, which we've seen a lot, right? So be familiar with that. They also have chemoreceptors and tentacles and photoreceptors that help them orient themselves in their environment and also sense what's in their environment and hunt and things like that. In addition to all of this, they also have some cells that are called neurosecretory cells. And these are nerve cells that produce and excrete hormones. And these hormones are important because they play a role in reproduction, secondary sex characteristics, and regeneration when needed. And then finally, they have these giant nerve fibers in their body that help them to move swiftly when they need to escape predators. And so you can see those giant nerve fibers here in this image. And they basically just help because if you stimulate that nerve fibers there, you can help stimulate those muscles very quickly and move a lot faster away from predators than if you didn't have those fibers. Annelids can undergo either sexual or asexual reproduction depending on the species and the particular environmental conditions. In the case of asexual reproduction, they can use something called transverse fission. And that's essentially where a posterior region of the animal will break off or bud off and become its own separate organism. So an example would be, let's say we have a worm, and this last portion of the worm breaks off or buds off and becomes its own separate worm. Now we have two worms that are genetically identical to one another, so they're clones of the parent. In annelids, they can also undergo sexual reproduction, and sexual reproduction in polychaetes and oligochaetes is a little bit different, so I've split this portion of the lecture into two parts. In the case of polychaetes, they are dioecious, so they have both male and female sexes. And in polychaetes, they also have temporal sex organs. So under normal circumstances, when they're not in mating season, essentially, their sex organs are reduced and they're non-functional. But once they're ready to undergo mating, their sex organs will become more developed and they will begin to swell. And then that swelling will allow them to become more pronounced and functional. And so they will make the gametes as respectively. So in the females, they'll make their eggs, and in the males, they'll make the sperm. And then the gametes are released into the coelomic cavity. Polychaetes are undergo external fertilization, so they must release their gametes into the environment and then hope that they meet with the corresponding gametes of the same species out there in the environment. So for example, one polychaete will release its eggs into the water, and then it hopes that its eggs meet up with the sperm of the same species that's also traveling in the same water. The gametes that are in the coelomic cavity must be released into the environment, and there are a couple of ways that a polychaete can release their gametes. One, they can make use of the metanephridia, and so this process works via kind of like this. So looking at this diagram, all these little circles here are the eggs in the coelomic cavity. And so the eggs will be swept up in the metanephridia and released into the environment via the gonoduct, sorry. And that's how they can be released. Another way that they can release their gametes into the environment is by just splitting open their body wall and releasing the contents of the coelomic cavity, so their egg and their sperm, out into the environment that way. Once the egg and the sperm meet and fertilization occurs, and then they start to the embryo starts to develop, they will have a trochophore larvae part of their life cycle. So polychaetes do have a trochophore larvae. This is different from what we'll see in oligochaetes. Sexual reproduction in oligochaetes is very different from what we see in polychaetes. For one, oligochaetes are monoecious, meaning that they have both the male and female reproductive structures and male and female gametes in the same organism. These structures are spatially separated, so you can see them in this image here. If you can't see it clearly, please go to your book or go to the internet to try to find a slightly better picture or a bigger picture. But they have their ovaries and their testes that are nearby to each other, and they have something else called a seminal receptacle. And the seminal receptacle is responsible for essentially storing the sperm from their mate until they're ready to undergo fertilization. And then in the case of the ovary for the eggs are produced by the ovary and then put into the coelomic cavity. And then once the organism is ready to undergo fertilization, they will release their eggs from the coelomic cavity through the genital pore into the environment. But this is not the same as what we saw in polychaetes. In polychaetes, they release their eggs into the environment to hopefully meet with sperm that's also present in the water column in the environment. In the case of oligochaetes, the egg is kept close to the body and is basically swept up in the cocoon that will later form that kind of protective coating for the eggs to undergo fertilization and for the juveniles to grow. Just as a reminder, that cocoon is produced via the clitellum, and the clitellum is only present in worms that are mature. So when they first hatch, when they first come out of the cocoon, worms do not have a clitellum. But once they're mature and they're sexually mature, they will develop this clitellum, and that will allow for them to undergo sexual reproduction. And this clitellum is always present in oligochaetes. In leeches, they only have it when they're ready to undergo reproduction, but in oligochaetes, it's always present. And that's important because they can undergo reproduction pretty much any time they find a suitable mate. So as long as the conditions outside are warm and it's moist, they find a good mate of the same species, they can undergo reproduction. So going through this process of reproduction and how this all works is we start with one worm. Let's say we're looking at earthworm in a burrow, and then another earthworm kind of comes in and they want to reproduce with each other. Both of these worms will line up along their ventral sides, head to foot. Well, not foot, anterior to posterior. So the head of one worm will be lined up with the posterior of another worm, kind of thing, like in those directions. And so they'll line up so that their reproductive structures are in just the right spot for them to exchange sperm. Once their ventral sides are lined up, the clitellum will be producing mucus, and that will help them basically stick together, anchor them together. They can also use their setae to help anchor them together as well, so they can reproduce. Once they're in place and everything is great, each of the worms will release sperm from its testes, and that sperm will be funneled into the seminal receptacle of its. The seminal receptacle will then store that sperm for fertilization later. So once all the stuff is happening, once the sperm is exchanged, the clitellum will produce a mucousy kind of layer, and that will be formed here. And that mucousy layer is then pushed anterior to the organism, and to be eventually pushed off of the organism's head, basically. So as this mucus is being pushed forward and forward, it will go past the oviduct and past the seminal receptacle. And as it's passing the oviducted seminal receptacle, it will pick up the eggs that have been released from the coelomic cavity, and it'll also pick up the sperm from its mate. And as the mucus is continuing to move down the organism, the eggs and the sperm will be mixed together to allow for fertilization to occur. Eventually, the mucus will reach the very end of the organism and be pushed off. As it's being removed, the ends of the cocoon are pinched off, or they're closed. So you get this nice lemon-shaped ball of mucus, essentially, that looks like this. And it's full of fertilized eggs. And then that cocoon will hang out in the soil for as long as time is needed for the fertilized eggs to develop into juveniles. And then once the juveniles, which are basically just very small, immature versions of their parents, of the adult worms, will leave the cocoon and then travel wherever they need to go. Alrighty, with that said, we have made it to the end of our lecture on annelids. You go ahead and read chapter 17, read the introduction, and then 17.1 through 17.4. And really, when you're reading through this section, focus on the form, function, organs, and reproduction sections of the of each kind of section. Okay. At the end, they have a lot of information on different worms, but really just focus on these big picture ideas for each of these types. And then the differences that I mentioned in this presentation, make sure you know those things as well. And then also do the chapter 17 Smart Book assignment. I've also included some videos here to help you to understand the material like I generally do. We have a video of an earthworm crawling, and so this is what I was talking about. If you're not, I know some people have probably never really seen a worm, or it's been a long time since you've seen a worm. If you need a refresher, if you need to know what I'm talking about when they're crawling, please watch that video. I have two videos here of fan worms, what they look like, and then the one on how they retract. It's really kind of cool. A video on the circulatory system in worms, and then two on leeches, so leeches feeding on other organisms. So with that said, you've made it to the end. See you in the next lecture where we talk about nematodes, or our roundworms.